Kumar VK, Dean SCTIMST. Our chief guest for this day, Dr. Rakesh K. Mishra, Director CSIR, Center for Cellular and Molecular Biology, Hyderabad. Would be joining online Dr. Monan PV, Associate Dean PhD Program, Dr. Santosh Kumar V, Registrar SCTIMST, to occupy their seats on the dais.
Check.
I request you all to stand for invocation.
സർവജീവ പ്രകാശമാകുന്ന സച്ചിദാനന്ദ ജ്ഞാനമേ ഹർഷഭാവന ലോലതന്തുവിൽ തൂം സാരസ്വഗന്ധമേ നിത്യവും ധർമ്മനീതി കാക്കുന്ന വിശ്വരക്ഷകരൂപമേ വിശ്വരക്ഷകരൂപമേ സർവജീവ പ്രകാശമാവുന്ന സച്ചിദാനന്ദ ജ്ഞാനമേ നിത്യവും നിന്റെ പാദപങ്കജം തൊട്ടു വന്ദിച്ചു വാഴ്ത്തുന്നു നിത്യവും നിന്റെ പാദപങ്കജം തൊട്ടു വന്ദിച്ചു വാഴ്ത്തുന്നു.
Honorable guests and my dear colleagues, we are gathered here today to celebrate National Science Day. National Science Day is celebrated on February 28 every year across India. This day is celebrated to mark the discovery of the phenomenon of Raman effect by Sir C.V. Raman. This remarkable discovery brought him the Nobel Prize in 1930, the first time for an Indian. On this day, we also recall and celebrate
to see the facilities and to have academic and research interactions with the institutions. So to meet that objective, because 28th February was holiday, to meet the objective, we decided to conduct our National Science Day. We are proud that this year we got brilliant, excellent students from our government women's college, Trivandrum. They are belonging to biochemistry, microbiology, and physics as well as chemistry. I heard. I welcome on behalf of this Department of Science and Technology. Every year we used to be the theme for the celebration. This year theme is future of science, technology, and innovation. And this focus on education, skills, and work. So we will get a detailed idea, detailed talk about this theme and science day by our respected Dr. R.K. Misraji from SCTIMST, respected director, Center for Senior and Original Director, CMD Hyderabad. On behalf of Sri Fraternity, I welcome you join online today. I welcome sir to this function, sir. R.K. Misraji, welcome to this function, sir. With, as you know, we are having 3 days. One is our hospital week, where all type of cardiac and neurology and another one is there in the same campus we are having Achuthananam Center. The public health related research and academic activities are going on. Third thing is where we are sitting here is our biomedical technology week. Here, medical device development as well as technology development is being used. So these things are going on under the supervision of our respected head DMPK. He is a material scientist and administrator of Sri Chitra Fraternity Group. Case investigated for 2 minutes.
Take maximum benefit out of what is being showcased today for you. And we are going to give you a science day message through one of our leading scientists, Dr. Rakesh Mishra, who is the director of CSIR Hyderabad. You are all welcome. We can have discussions with our scientists here, and you are welcome anytime. And we are also having programs like internship and observership for students who are interested in this field. They can come and spend more time with us. And the ultimate aim is to get more and more people interested in science and research.
Thank you, sir, for sharing your wonderful and inspirational thoughts. The theme for this year's National Science Day is the future of STI, impact on education, skills, and work. The theme is how science, technology, innovation would impact education, skills, and work in the future. Let me begin with a quote from Dr. R.A. Marshallkar, renowned scientist and intellectual, quote in quote. India needs science that solves, technology that transforms, and innovation that impacts. The pandemic has made the nation realize the importance of science, technology, innovation as a savior. To tackle present challenges, the Government of India has come up with India's fifth National Science, Technology, Innovation Policy, which aims to capture the aspiration of entire nation through decentralized, bottom-up, and inclusive approach. Policy outlines the future of science, technology, innovation under following broad vision. The first, to achieve the technological self-reliance and position India among the top 3 scientific superpowers in the decade to come. To attract, nurture, strengthen, and retain critical human capital through a people-centric science, technology, innovation ecosystem. To double the number of full-time equivalent researchers, gross domestic expenditure on R&D, also called GERD, and private sector contribution to GERD every 5 years. To build individual and institutional excellence in the science, technology, innovation with the aspiration to achieve the highest level of global recognition and awards in the coming decade. The science, technology, innovation vision is a step to build a strong foundation for the future and achieve the goal of Atmanirbhar Bharat.
Today, let's hear words of wisdom from Dr. Rakesh Mishra, Indian scientist in genomics and epigenetics. He is the director of CSIR Hyderabad. He had obtained MSc Chemistry in 1981, PhD Organic Chemistry in 1986 from the University of Allahabad, postdoctoral research on non-BDNA conformations and DNA topology at Molecular Biophysics Unit, Indian Institute of Science, Bangalore. He worked at Center for Cellular and Molecular Biology, Hyderabad as scientist and studied transcription initiation in prokaryotes, chemical recognition, and cleavage of DNA. 1996 to 2001, he worked on chromatin organization and regulation of homeotic gene complex of Drosophila melanogaster at the University of Geneva, Switzerland. He is also an elected fellow of J.C. Bose Fellowship, Indian Academy of Sciences, National Academy of Sciences India, Indian National Academy of Sciences, Andhra Pradesh Academy of Sciences. He has published more than 100 articles in national and international journals. I invite Dr. Rakesh K. Mishra to deliver the Science Day message.
Thank you. Thank you very much for inviting me. It's really a privilege and honor for me to be with all of you, although in virtual mode. I would like to try and share my PowerPoint and hope it works. Is the PowerPoint visible and I'm audible? Somebody can signal?
Yes, sir. We can see you.
Okay. Okay, perfect. Great. Yeah. Thank you. So I'm going to describe very briefly some aspects of science in the context of pandemic and what academic institutions can do. And maybe some 15-20 minutes. And I would like to then, if there are questions and any clarifications or discussions, I'll be very happy to do that. So, as you just heard, that this pandemic has changed many things in our life, and it has brought to the front the science and technology. Because when there are unknowns, then all we need is to do research, study more, and figure out what is happening. Um, right. So the COVID-19 had been the worst public health crisis in a century. We all know now. In past 1 year, 100 million more people have been infected all over the world. 2.5 million and more deaths, and still is counting. And it has disrupted the life, public life, and the way we do our regular things and the economy in an unprecedented manner. While that has happened, it has also catalyzed the research, a revolution to understand the virus, which didn't even have a name about a year. So I'm going to put some context that institution like CCMB that is mandated to do fundamental research of the biology, how modified its approach in the interest of the requirement of the moment, and responded to this crisis in certain ways that I will use as a example to highlight how academic institutions and scientists and students can contribute to society in a major way, and how these will have bearing on the policy of education and science both. So the pandemic of this kind is once in a lifetime opportunity or challenge, whichever way you take it, where scientists can make a difference. For example, help the healthcare system and hospitals when new things have to be brought into the place. Understand the biology of the virus so that we can tackle the situation, find interventions, and participate in different programs. All academic institutions can do it. For instance, participating in repurposing of the drugs or new drugs, traditional interventions, very important, and the vaccines. We can participate in many different ways. And since it is a new kind of problem, we have to find new solutions and emerging aspects of the problem, because it's changing its nature as time passes. So it's like a changing goalpost. So when an infection or a disease of this kind arrives, what we need to do immediately? So immediately we need detection and diagnosis. For a country like India of this size, it should be accessible, it should be feasible, and it should be high throughput method of diagnosis and detection. Because unless we are able to detect, we will not understand what is happening, where is the disease, and how fast it is happening. Then there is a way to control the spread, and this can be done in multiple, there are multiple options. So technical interventions, and behavioral intervention and government policy intervention, they all are needed with proper information backing in order to control the spread. And the associated issues of this kind of are new normals, what we call the information and policy and ideas, how to handle this problem which we didn't even have any idea only a few months back. So considering the scientific and technical context, academic organizations must take initiative, innovate, and contribute to the problem, because no one else will be able to help in this. Only scientists and people who can understand the technicalities can come forward. And one more important thing, extremely important thing, is communicating the unknown. Keeping the politicians, the policy makers, academic peers, media, and general public informed is very important, because if you don't give credible information, then people will be fed with the rumors, and that will lead to chaos. In times of uncertainty, it becomes extremely important for people to have credible source of information, and scientists hold a very important role during a crisis like this, and hence it has been more important than ever to be accessible. Scientists being accessible to people, to press, to media is extremely important, because we can give the correct perspective. Also, the new information is adding every few days. So it is important to have a continued discussion with media. It's not that you release one press note or some information sheet and that is enough. The virus is changing, the disease pattern is changing, new things are happening. So we need constant discussion with the policymakers and the public to give them the best suggestion at that time. So remember, even medical doctors don't know much about this virus which they are dealing with, and therefore it becomes a duty of the scientists to do fill the information gap. In this context, I will bring you few of the things which CCMB has been doing, which will give you a perspective of how we can respond to the situations like this. And before I go further, I would like to say that all I'm going to talk, more than 90% what I'm going to talk, are the initiative of our students. Students means we have PhD students, about 150 of them. It is their initiative, is their brave approach towards this kind of problem that we have been able to respond to the disease to the degree that you will get to know a glimpse of that in a few minutes. Because of these initiatives are taken by students, we established a testing center for the COVID, we have a training center, the validation for new technologies, new tests, and the national repository of the virus and patient samples. These were the activities, they are all recognized by ICMR. In the meantime, we also started culturing the virus in the lab, which was very, very useful, because we as well, and we had required cell lines and media and so on. So we got the patient samples and we started to culture this virus, which opened many avenues to handle the and possible interventions to be developed. Like, for example, you can do testing and validation of devices. Also, when the young students, creative is working in something, they come up with the new suggestions, new ideas, new improvements, and we ended up developing many diagnostic methods and protocols and SOPs. We also followed the genome of the virus, how it is varying, because that's expertise of this institution, and all these things have helped in understanding the biology of the virus. So I will give you some examples, and all the pictures you will see individual, they are all PhD students. So you can imagine that what you all can do in this time of crisis. So as I said, it's not necessary to be working in the BSL-3 labs. You can also read and share correct information, which is very, very helpful for the people. So when this crisis began in the end of the March, at least in India, when we were told that this is going to be a big difficulty, we were called by the state health department, and we had a meeting with the top officials there. And we worked out a strategy in the how CCMB is going to train the medical staff first, because we have all the facilities, and then we will start also testing the virus itself in our own labs. And so the first thing was to train all the 5 medical institutions here, their staff, to how to handle safely the samples, and then interact with them. Some little supplies they don't have, help with that. And it was very nice relationship that we built, because they felt very confident after seeing that we are handling the virus like this. it was very, very important, very useful. We also started testing. We were recognized by ICMR to do testing. And in this process, we made many improvements like reducing the VTM, pooling the samples, and saving the time and energy and reagents to get more tests done in the same time. We also developed new methods of testing. Two of them are listed here. One is the nested PCR method for surveillance, which is extremely sensitive method, and it can be used for community surveillance purposes. And then dry swab method, which I think is very, very important, as you will see in a minute. So normally for testing, many of you may have experienced it, or many of you have seen it, that a swab is collected from nose or throat, and this put in this pink solution, which is called viral transfer medium. And then that is packed nicely because it can leak, and then it comes, and then it has to be carefully unpacked in a BSL-2 conditions where the technician will be wearing double gloves and so on. And then you go to RNA isolation and then do RT-PCR for the reporting. So extremely cumbersome process. So what we means our students, what they did was they worked out that you do swab collection, but don't put in this pink tube, just send the dry swab to the lab. Unpacking is very easy then because it's just dry swab you are taking out of the tube. There's no risk involved, no leakage. If it's anyone, if you have experience, you know when samples come, how messy it is. Leaking samples and with the viral infections is a very, very big hazard. And then we also figured out that there is no need to do RNA extraction. Simple incubation with the simple buffer, very cheap buffer is enough to go for RT-PCR test. So when now this, by the way, this method is now approved by ICMR and can be used by anybody. It uses less time, half the time, less cost, and you can, with the same setup that you have in the lab, you can have 3 to 4-fold higher throughput. So we think that it will be a crucial thing which is already in place in many institutions. We have trained labs in Hyderabad, in Nagpur, in Chennai to use these things, and we have even signed agreement with Apollo Hospitals, Spice Health, and Merrill Diagnostic. They will so Spice Health is using it in Delhi in their mobile vans. Apollo Hospitals has developed a kit which is being validated in Chennai and should be out soon. Merrill Diagnostics has also approached ICMR with the version of this kit because it will be cheap and safe to use that. So this contribution of young students who were just working around doing the test and all the time thinking and changing the working environment completely when you have to when you are working safer, smoother, and more efficient. I would like to bring another aspect of this disease, which is zoonotic transmission. So zoonoses are the infectious diseases transmitted from animals to humans and can evolve to become efficiently transmissible human to human infections. So current pandemic, as we all know, is thought to have originated from the wildlife food market in China's Wuhan city with probable origin from a bat-borne virus. So pathogens that cause outbreak in humans evolve in reservoir hosts such as rodents, bats, or small mammals. Transmission of SARS-CoV-2 from humans to Minsk in Europe and US can create such reservoirs, and increasing danger of zoonotic transmission as humans come into greater conflict with their environment. So all I'm trying to see is that we are interfering with the nature, interfering with the natural environment of animals who are closer to us. That means they are mammals, and they can also share the virus at least some time and become a reservoir and transmit to people. And to begin with, also, like, we can bring the virus from the wild to the market and then spread to the people. So this is extremely important thing that, so before we go that, so I just want to bring to you that there are clear examples of, like April 1st, the first case of COVID was seen in the animals. India got in Bronx Zoo when the tiger died. Then India got activated, and on 20th July, I will show you a picture of this sample, which was tested in CCMB. And after that, many studies have been done that there are at least 13 species out of 58 that are tested that can actually transmit the COVID-19. And as one would expect, old world primates, mustelids, felids, and canids are the more efficient in this process. And as some of you may have heard, in Spain, uh, 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 some lions got developed this infection when the zoo staff was positive. So when a zookeeper in Hyderabad Zoo was positive, a lion became sick, and then we were then facing the challenge of how to test the COVID level in the lion. So we worked with Central Zoo Authority, with the zoo keepers, and worked out how to collect the samples, how to test them, do proper controls, and of course this problem was solved, and the lioness was negative for the COVID in this case. But we are now working in close collaboration with Central Zoo Authority to testing our animals whenever there is suspicion, and making sure that they don't become infected or become a reservoir of virus to transmit it in newer version to us again. Is I mean, one of the important message I would like younger people particularly to know, that if we wildlife is life for us as well. If we don't respect it, if we treat the animals indiscriminately like minsk and pendolins, if we eat wildlife without proper rules and guidelines, like people have done in Wuhan, and if we destroy the forest, if you take their home away, they will come to where we are, more closer to us. So to stop future pandemics, we have to do many things. And one of the most important thing will be to share the nature, to share, to respect the nature, respect the wildlife, because we have very little understanding of how we are connected in this ecosystem. And if one component has been taken away, what kind of disabilities it will bring in our functionalities, we don't have proper understanding. And so therefore we should be respectful to our environment. Uh, so with that, and we are continuously doing this, we are having a large program of zoonotic survey of viral and other infection loads, which we study through the ticks that come to cattle, or they can be collected in the forest floors. One of the important things we want to do in the times of pandemic is to involve industry, because government systems can do only that much. Only when the private players and industry come in picture, we can make really the changes. And therefore, we need to help and support innovation and self-sufficiency. As you just heard, India is putting a lot of emphasis, and rightly so, to be self-sufficient in these processes. So one thing which we began, as I mentioned, was to grow the virus in the lab. And if you culture the virus in the lab, what does it do? One is, now you have virus growing in the lab, so you can do experiments and you can understand the biology of the virus, which you can't do in a patient. And you create a platform for testing, because now you have virus growing in the culture, so you can see what prohibits this growth. So those will be potential drugs. You can see what kills this, what kind of not edible or not drug, what kind of methods can clean. So you can work out new sanitization strategies. And you can develop neutralization assays in which you can check the antibody or vaccine serum, people who have been given vaccine, if they are having neutralization antibodies. So all these become available when you have the cell culture of the virus going. And of course, you can use this to develop anti-sera or vaccines. And we are involved in all these processes. We are growing 4 variants of this virus culture, including the UK variant, which came recently, so that we can test appropriate drugs and vaccine. We have held a number of industries. Now, this is a little bit older. We have about 120-30 industry products and academic institution products which we have tested and given the results. And many of them are extremely important, extremely exciting. And some of them include, since it may be relevant to many of you, from Kottayam, and the big Ayurveda Institute, we have tied up with as an MOU, and we are testing the many herbal products, and some of them have given very interesting results. And we want to put this, bring a traditional knowledge into realm of in the domain of modern science, so that it can achieve its due status in the society and in the business arena. And this is one way we can when we can begin, which we have already done. It will not be limited to pandemic. It will be expand to many other infectious and other things. One of the strategies as a government policy and as our responsibility of taken is in the capacity building in the diagnostics. So we have created a consortium of industry partners who are making the components of the COVID diagnostics. And the idea is to encourage them, provide them technology when they make the components, assess their quality, scalability, and compatibility in the RT-PCR based diagnostic in this particular case, which can be generalized later. And we have set up a network of suppliers and manufacturers so that we can internally source all the components and assess them in-house. The target is in coming few weeks, we will be able to say that no component is now required for import for doing these kinds of activities. And we are focusing both on antigen and antibody-based rapid diagnostics, including also, as I mentioned, RT-PCR. And we are also assisting in developing diagnostic kits with developers, with advice and pre-validation testing and so on. So the idea is that if we become self-sufficient in a diagnostic kit is one thing, but research components, if they are available, then in smaller universities and colleges, things can be easily made available, and they can do quality experimental projects and research, which is very minimal in our colleges and in many universities. Research is limited to very few institutions, elite institutions like yours, and not in the many colleges. So we think that this kinds of indigenization, when things become easily available, we are not depending on import, it will help, it will give a boost to research activities in these colleges and attract young people to come to science, which is going to be the really crucial thing. If we want to be developed and a strong nation, we must develop our science base. As the previous speaker mentioned, we must spend more money, and all this is linked. If you do quality science, you come up with the innovative results, people, public, government, and most importantly, private sector get interested and spend more money. It will not happen that you just keep waiting till you get more money, then you do something. So it's a catch-22. So therefore, we should begin with what we have and then expect others to chip in. This is one of the examples. I will not go in detail, it's technical thing. So, but one of our incubators. So we have an incubation center in CCMB. We call it Atal Incubation Center because it's funded by Niti Aayog. And there, one of our young entrepreneurs startup has developed a method to detect antibody against this virus using Octet, this biolayer interferometry BLI platform. Some of you may know. This interesting thing is that this test can be done in less than a minute, and you can do 50 rupees for one test, and you can do 200 tests in an hour. So you can do large scale testing of antibodies using this, and this is being now assessed by ICMR for formal approvals. We have interacted with another company, Syngene, which is a biocon company. And in this, I will just give you a summary that this technology that we are more or less now completed with the Syngene is a technique in which you can do 50,000 up to 50,000 samples in one go. In which, and one go means one batch takes about 2 hours or so, 25, 30 hours, maybe a little bit more, 36 hours, but you can do 50,000 tests, and the cost will be 200 rupees per test. So which means you can test the entire battalion, entire school, or entire industry, entire mohalla using this in one go. So these are things which have our students have suggested a thought, because they are using these things in their research. Now they have put their brain and come up with these kinds of innovative methods. The other component of this pandemic is the genomic surveillance. As you are hearing, the new variants are coming, and we can do by genomic surveillance. You can detect them, because in fact that's the only way you can detect them. And the tracing the virus, how it is moving around, and how to define new strategies, new strategic moves. We need to know those kind of information. As you have seen that monitoring the virus spread is extremely important, and we see so much of variation from person to person, so many people asymptomatic. So why this is so much different? Is the people genome that is different, or virus is different, and so on? So we have been doing from the beginning very extensive analysis. Recently it has been made public, most comprehensive 1 year history of this virus in the country. So, and just to some of you who are interested can go to this website. This is CCMB app that we have produced as a Gear 19. Just type Gear 19 CCMB. You can go and play with this app, and you can get all this information played. How many clades are there? The 5,800 samples have been sequenced all over the place from 35 labs, 21 states, and so on. Interesting thing is more than 7,000 variants we have cataloged. So that means that's the dynamics of this virus. And these are all the states you can see the coverage, which means the virus is, most of these variants will have no consequence. So they will never be seen again, but some of them may become nasty. So we have to constantly monitor it. And one of the example is this. Different color means different clades or different kinds of variants of this virus. As you can see in the beginning, lots of different clades were available, and now you have only one of them. But we have to keep following, because this scenario keeps changing and new variants keep emerging. So this kinds of analysis keeps us at the front of what is happening in the virus. So some of you may be interested. You may have heard now the spike protein, receptor binding domain, many things. So here I'm just wanting to show to you, to cautious you that we are not quite done with this pandemic yet. These are the new mutations that have been emerging in recent past. As this green thing, what you see is in fact the most crucial thing of this virus. This is the part of the protein which is sitting on the surface of the virus, the spike protein, and binds to the receptor on the human cell in the respiratory system and gets the entry into the cell. And as you can see, there are so many mutations that are known, not in one line, but like, for example, this is the UK variant that you are seeing. This is this is very efficient in spreading, but doesn't cause worse disease. This mutation E484K is a South Africa and Brazil component, which is suspected to even escape the immune system. This 440K, which is in this part of this protein, is actually spreading quite significantly in south of India, including Andhra Pradesh, Telangana, Kerala. Close to 40% is this. Luckily, this is not escaping the immune system yet, and it's not causing worse form of disease, but the landscape is changing of this virus. So that therefore we have to be in constant watch, and towards the end I will just give 1 or 2 slides in which I will make my suggestions what we do. Before that, I just want to spend 2 minutes in 2 other surveillance that we are doing. One is air surveillance, because we know the virus spreads from person to person through the air. So we set up experiments to collect the air sample from various context, whether it's COVID ward, COVID ICUs, various parts of the virus, and even some places of people, apartments where who were willing to volunteer. These are our students, all geared up in the hospital context. And instead of going to detail, I'll just give you this quick summary of what we have seen. So in a closed environment where there are many patients, you can have the virus as far as 20 feet from where the patient is, if the patient is emitting enough viruses, right, depending on other conditions. In a closed room, there's a lot of virus in the air and it can hang around for some time in dining hall, bedroom, anywhere. And we have noticed in the toilets, virus can stay up to 2 hours or even more. And the reason for that is when people go to toilet, they remove the mask. So when you go, whether you have infection or not, when you go to toilet, public toilet particularly, must wear mask. Don't think that nobody's around, so you are safe. You are most vulnerable there. Because even if 2 hours back somebody has used the toilet, the virus is still in there. So this, we have made advisory and shared with people, and anybody interested will be very happy to share that. If people are infected and talking on phone, there is lots of virus in there. If people are wearing mask when talking or on phone or general conversation, virus is not detectable, and so on. So many. So the key thing is outdoor air is largely free of the virus, so we don't have to panic and worry about it. So lots of depend on the situation, but in closed room environment, you must, and when there is somebody coming from outside, you must wear mask, and wear mask even after half an hour after the person has left, unless there is enough airflow and windows open and fan on and so on. So that is something which we should do. The other thing we have done is surveying the sewage system. See, all the discards from the toilets, from the sink, from the bathrooms, they end up in the sewage system. So we developed a technology with the help of our sister laboratories, IICT and NEERI in Nagpur, to collect the sewage samples and develop technology of calculating the virus load in the sewage system. And then depending on the volume of the virus and how many people sample come there and number of what the population in the area, we can actually tell now very confidently what is the percent of people infected in this part of the city. So we are monitoring for several months now, 10 different cities, the viral load, and this matches excellently with the other methods of which are less accurate because they are based on extrapolation of the data, and one I will show you in a bit. So this is a very cheap, very convenient and efficient and accurate way of telling the viral load. And again, all this was done by our PhD students who took initiative, and we just gave them free hand. And this is the last part of survey. We have done the zero survey. Result will come this week in public. So I can share with you that this has been done with in collaboration with NIN. They have expertise of sample collection and Bharat Biotech that helped in this study and supported it financially. So with the survey of 10,000 samples that we have done, based on that, we can say that in Hyderabad has close to 60% seropositivity. That means these many people have antibody against this virus, which means maybe 70-80% people have gone through the infection, right? So that's amazing thing. So now last 1 minute, I would like to just say that what is the current situation. Now I gave you something what we have been doing in whatever we could do, some aspects of those. I didn't tell you what basic research we have done on this virus, so that is not of much interest for the general audience. So the pandemic status in India at the present is that number of cases are declining from the beginning of this year. But we all are now, and we have been constantly emphasizing, we are not yet done with it. Although you see the death is decreasing, the curve is plateauing, the number of new cases, a lot of people have recovered, but we are still there in the danger zone because of this table you see. These are the new variants that are origin of this country, and what many of them have already landed in India. And remember, we can also make our own variant. If more than 1 lakh people are infected, virus is mutating. As I told you, more than 7,000 mutations we have cataloged. So if we are letting the virus do its job, then we are creating in future new variants unknowingly, which can lead to severe problems. So what we can do, vaccine is a big tool for us. So, and this has been really like a miracle. We can discuss even hours, the vaccine story, very fascinating. Interestingly, never before, 2 weeks prior to the first anniversary of COVID-19 detection in the country, which was done in the state of Kerala, where you are sitting, new vaccine was launched in India. It has never happened that within a year, vaccine is available. And so far, crore of people have been already vaccinated. Some have finished the full dose of vaccination, the two dose. And in 2 days back, this is the data. And from today, it is going to be really accelerating because now it's available to public in the of certain restriction, of course. But most importantly, the private players are now involved. So we will be now maybe vaccinating uh, uh, up to 500,000 people in a day soon, which is required. It is required, but we must also keep in mind that vaccine is not immediate protection, or the news of the vaccine is not a protection. Even if you are vaccinated, there is chance that 10-15% of us will not get protection. And even if we are going to get protection, it takes 45 days to get the full protection. So even if you are vaccinated, you must use this, the social vaccine, which is the proper mask. And I think uh, people like you should be example for the society, that shows that how to wear a mask, how to be disciplined, how not to cluster together, and how to keep hand wash. These are general practice, which is not only for pandemic, it can be useful for many other things. But for the moment, next few months, I think I urge all of you to spread this message that vaccine is not the solution immediately. It is a good tool. This we can be, it's very good tool has come at the right time when we have upper hand on the virus, spread is less. But if we don't do these methods, then we will lose this war, and one more pandemic will not be manageable. It will be too much to bear. It's just simply unaffordable. So we have a responsibility as an academic organization, as students of science, as researchers, that in future we should be prepared. We should be doing constant surveillance of the vectors like mosquitoes and flies, and the parasites like viruses, protozoans, bacteria. There are so many of them. I already discussed zoonotic disease surveillance, how important it is, and it is possible to keep a track on this by zoonotic surveillance, and hopefully preempt a future pandemic, because one pandemic takes really the breath out of us from our economy. And we have now learned lessons to be indigenize, to indigenize the things that we need, quick adaptation, because new disease will come, so that will require adaptation, and we should be prepared with technical and scientific power and setup. I mean, CCMB had BSL-3 and all this setup, so that we could, within a week, within few days, we could set it up as a diagnostic center and a virus culture center. So that preparedness is important. And one good thing from this pandemic is that all these aspects have now been highlighted, and I hope that we are getting better prepared for the future in this context. So with that, I will thank you. This thank you slide has these pictures, because this is how we are functioning now. Everybody enters the campus only after temperature is checked. After entering, you wash your hand, and we have many places. What are the rules and norms follow? Always wear mask. And I can tell you, for 1 year, we have not closed our campus even for one day. It's all the students, hundreds of them are there. They eat in canteen. We follow COVID discipline, and we have not seen even one outbreak till now, though we have tested 70 80,000 samples proving virus in the culture. So this I want to say is that people like you can set up an example, like our students have said, that there are ways of functioning even in this environment without letting the virus exploit us. If we protect ourselves and follow certain discipline, we can manage this disease very well. So the social vaccine is going to be more crucial than any other vaccine. Other normal vaccine is a good thing, but it will take its time, and we will be able to manage only if we are following this COVID appropriate behavior. Otherwise, even a vaccine may fail if a variant of a nasty kind emerge. So with that, I'll thank you all for your attention, and I'll be happy uh, uh, to answer any question if there are any. So thank you all, and I try to
for sharing magnificent details on SARS-CoV-2 and use of science, technology, innovation to detect, in surveillance, and contain this contagious virus. Now I invite Dr. Harikrishna Verma, Head BMT Wing, to offer felicitations.
Respected Director Professor Kumar, respected Rakesh Mishra for your my Dean, Dr. Ajit Kumar, and dear students and teachers. ഞാൻ പറയാൻ തുടങ്ങാൻ വന്നത് ചില കണ്ടുപിടുത്തങ്ങൾ വളരെ വളരെ കാലാതിവർത്തിയാണ്. നമുക്ക് എത്ര കാലം കഴിയുമ്പോഴും നിങ്ങളെ പോലെ കെമിസ്ട്രി സ്റ്റുഡൻസിന് അറിയാം, വളരെ ചെറിയൊരു മോളിക്കുള്ള ഗ്രിഗ്നാർഡ് റീജിയൻ RMGX, അത് നമ്മുടെ സിന്തറ്റിക് കെമിസ്ട്രിക്കും അതുപോലെ തന്നെ എത്രയെത്ര കെമിക്കൽ പ്രൊഡക്ഷൻസിനെ ഹെൽപ്പ് ചെയ്തു. അതുപോലെ തന്നെ നമ്മുടെ മഹാനായ ഇന്ത്യയിലെ ഏറ്റവും വലിയ ശാസ്ത്രജ്ഞനായിരുന്ന നമ്മുടെ CV രാമൻ്റെ രാമനെ ആദ്യമൊന്നും അത്രമാത്രം കണ്ടുപിടുത്തങ്ങൾ അതിൻ്റെ പുറത്ത് ഉണ്ടായിരുന്നെങ്കിൽ നിങ്ങൾക്കറിയാം, ലാസ്റ്റ് 10-20 വർഷങ്ങളായിട്ട് ഒരു വർഷവും ഒരു സ്പെക്ട്രോമീറ്റർ, രാമൻ സ്പെക്ട്രോമീറ്റർ ഉണ്ടായിക്കൊണ്ടിരിക്കുന്ന സമയം. കാരണം നമുക്കറിയാം സർഫസ് എൻഹാൻസ്ഡ് രാമൻ സ്പെക്ട്രോസ്കോപ്പി, അങ്ങനെ ഓരോന്നോരോന്നും. ഏറ്റവും ഒടുവിലായിട്ട് ഇപ്പം മാർസ് റോവറിൽ പോയിട്ടില്ലേ, പെർസിവറൻസ് എന്ന് പറഞ്ഞാൽ, അതിനകത്ത് ഏറ്റവും പ്രധാനപ്പെട്ട ഒരു സ്പെക്ട്രോമീറ്റർ ഷെർലക് ഹോംസിന്റെ പേരുള്ളതുകൊണ്ടായിരിക്കും SHER. ആ ഷെർലക്കിലെ 6 ഒരു രാമൻ ആൻഡ് L ലുമിനിസെൻസിനാണ്. അപ്പോൾ ഈ ഷെർലക്കിന്റെ ഉദ്ദേശം എന്താണെന്ന് വെച്ച് കഴിഞ്ഞാൽ, മാർസിൽ ഏതെങ്കിലും ചെറിയ ഒരു ജീവന്റെ കണിക ഉണ്ടെങ്കിൽ അതുപോലും പിക്കിയെടുക്കുവാൻ ഈ 100 വർഷത്തിന് ശേഷവും അതിൻ്റെ രാമൻ എഫക്റ്റും അതിൻ്റെ കണ്ടുപിടുത്തത്തിൻ്റെ ഉപയോഗവും ഇപ്പോഴും നമുക്ക് പ്രസക്തമാണ്. അപ്പം ഈ ദിവസം നമ്മൾ ആഘോഷിക്കുമ്പോൾ തന്നെ നമ്മുടെ ഓരോരുത്തരുടെയും ശാസ്ത്ര ചിന്തകളും അത് മുന്നോട്ട് നമ്മുടെ മനുഷ്യ സമൂഹത്തിനെ എത്രമാത്രം പ്രയോജനപ്പെടുത്താൻ കൊണ്ടുപോകുന്നു പറ്റുമെന്നുള്ളതും ഒരു ക്ലാസിക് എക്സാമ്പിളാണ്. അതുകൊണ്ട് തന്നെയാണ് ഇത്തരം വലിയ വലിയ കണ്ടുപിടുത്തങ്ങൾ പിന്നെയും സമൂഹത്തിൽ എപ്പോഴും നമുക്ക് വേണ്ടി തന്നെ വീണ്ടും വീണ്ടും, ഉദാഹരണത്തിന് ഈ പറഞ്ഞ നമ്മുടെ പ്രൊഫസർ മിശ്ര പറഞ്ഞ പോലുള്ള ഈ ഡിറ്റക്ഷന് വേണ്ടിയിട്ടുള്ള കോവിഡ് പോലുള്ള പുതിയ പുതിയ രോഗങ്ങളുടെ ഡിറ്റക്ഷന് വേണ്ടിയുള്ള ഉപകരണങ്ങളും ഈ ഡയഗ്നോസ്റ്റിക്കുകളും ഈ സാങ്കേതിക വിദ്യ കൊണ്ട് തന്നെ, രാമൻ എഫക്റ്റ് കൊണ്ട് തന്നെ ഉള്ള പുതിയ മെഷീനുകൾ വരുന്നതും നമുക്ക് കാണാൻ കഴിയുന്നതാണ്. പിന്നെ ഒരു ചെറിയത്, നിങ്ങളെ എല്ലാവരെയും BMT വിങ്ങിലേക്ക് സ്വാഗതം ചെയ്യുന്നു. ഇവിടെ നിങ്ങൾക്ക് ഇത് തീർച്ചയായിട്ടും ബെഞ്ച് ടു ബെഡ്, അതായത് ലാബറട്ടറിയിൽ ഉണ്ടാക്കുന്ന സാധനങ്ങൾ അവസാനം എങ്ങനെ ഡോക്ടർമാർ അത് രോഗികളുടെ പ്രയോജനപ്രദമായ കാര്യങ്ങളൊക്കെ ചെയ്യുന്നു. അപ്പോൾ അതിനുള്ള കണ്ടുപിടുത്തങ്ങളും ടെസ്റ്റിങ്ങുകളും എല്ലാ കാര്യങ്ങളും നിങ്ങൾക്ക് ഒരു ദിവസം കൊണ്ട് തന്നെ ഇവിടെ നടന്ന് കാണാൻ പറ്റുന്നതാണ്. അപ്പോൾ ഈ ചെറിയ വാക്കുകളിലൂടെ ഒരിക്കൽ കൂടി ഈ ശാസ്ത്രബോധവും ശാസ്ത്രചിന്തയും നിങ്ങൾ വളർത്തട്ടെ എന്നും പുതിയ പുതിയ ശാസ്ത്രജ്ഞരായി വളരട്ടെ എന്ന് പറഞ്ഞുകൊണ്ട് നിർത്തുന്നു. നമസ്കാരം.
Thank you, sir, for encouraging words. Now I request audience, if there are any questions, sir is waiting for it. Any question from audience? Oh, no. Okay, sure. Thank you. So now I invite Dr. Ajit Kumar, VK Dean, SCTIMST, to share few words with us.
Good morning, everybody. Respected Director Dr. Jay Kumar, Director CSIR, CCMB Dr. Rakesh Mishra, Head BMT Wing Dr. Verma, Associate Dean Dr. Mohanan, Registrar, members of the faculty, and my dear friends. Good morning. It is indeed a privilege for me to address you youngsters. And I wouldn't dwell much on the significance as to how the Science Day has come into. You have been told about that. But then one should understand that this day is dedicated to the legendary scientist Dr. C.V. Raman and for his discovery of the Raman effect. And today, as I was reading Raman effect, most of you may not know as to why the sky is blue, or for that matter, why the ocean is blue, and how and why the ocean is blue is related to what he has explained. And if any of you do not know that, please go and read it and then find out why it is. If you look at the evolutionary biology, you can find out that there is no significant genetic difference between that of humans and mammals. Probably what has happened is a mutated or an evolved frontoparietal cortex of the humans have given us this edge and made us dominate over the species. But what I would like to tell you is that that does not make us have the best of hearts or the best of the immunological systems. And probably that's the reason why we are as much sensitive, or we are victims to this SARS-CoV virus. Now, coming to the future of what science would be, it's very difficult to make even a prophecy of how the science will be. I was looking at some of the things that one can think about as to what science will be in the next 10 years. I suppose you will have robotics, and you will have a robot in your home at the beck and call. You will have a car which will come on just a mere message without a driver. But if I were to tell you that cancer can get treated and that aging may slow down, you may not be impressed. But if I were to tell you that animals and plants can be manufactured, you may find it unpleasant. But then all of it is happening. So let us see what some of those science which happen day to day with us. I can tell you that, for example, in the world, you know that the messenger RNA is synthetically manufactured, injected, and then that is how we have evolved into vaccine development. But if any of you have an abnormal heart rhythm with you, I can tell you that I can map out the rhythm in your heart, map it in 3-dimensional, and then localize it to about 10 to 25 milliseconds within your heart. And then using radio frequency impulses, I can take care of that or ablate it or burn it off. So we have received so much. So there are a lot of discoveries that has happened or we do in a day-to-day practice that most of you may not know. And that is actually an eventual reality. So young friends, I should tell you that there is no end to dreaming. Please dream. Aim for the stars. You may get the moon, but you may actually get the stars itself. Thank you.
Thank you, sir, for enlightening us. Now it's time to propose vote of thanks. I would like to invite Dr. Mohanan PV, Associate Dean, PhD Program, to do this.
A very good morning to all. Respected Chief Guest, Dr. Rakesh K. Misra, Director, Center for Cellular and Molecular Biology, CCMB, Hyderabad. Respected Director, Professor K. Jayagumar. Respected Head, Biomedical Technology Wing, Dr. Harikrishna Verma. Respected Dean, Professor Ajit Kumar, VK. Registrar, Dr. Santosh Kumar, students and faculty members of Government Women's College, Trivandrum, and faculties and students of our institute, and dear friends. As you know, the National Science Day is celebrated in India on 20th February each year to mark the discovery of the Raman effect by Indian physicist C.V. Raman on 28th February 1928. For his discovery, Sri C.V. Raman was awarded the Nobel Prize in Physics in 1930. We are very happy that Dr. Rakesh Misra, Director CCMB, delivered the National Science Day message. In spite of his busy schedule, he accepted our invitation to deliver the National Science Day message. Sir, on behalf of all the staff, students of Sri Chitradinal Institute for Medical Science and Technology, I wholeheartedly thank you, sir, for accepting our request. Thank you, sir. I express my deep sense of gratitude to our director, Professor K. Jayagumar, for his encouragement and generous attitude throughout the organization of this event. Without his active contribution and support, this would not have come true, sir. Sir, on behalf of all the participants and on my own behalf, please accept our sincere thanks. I acknowledge my wholehearted thanks to Dr. Verma, Head Biomedical Technology Wing, Dean Professor Ajit Kumar, and Registrar Dr. Santosh Kumar. It is indeed a great pleasure to welcome you all and students and faculties of Government Women's College, Trivandrum, for their active participation in the National Science Day celebrations. My special thanks to the principal and Dr. Arun, Government Women's College, Trivandrum, for their outstanding support for organizing this event. Today, as a part of National Science Day celebration, we organize several programs such as a talk by Professor Harikrishnan, Professor of Cardiology, a science quiz by Dr. Ranjit S, and Science Magic by Jijo Raj, Dr. Jijo Raj. And after this, there will be laboratory visit. Thank you all for your kind support. I'm greatly thankful to all my colleagues. I'm not naming individually, and who are working hard for the grand success of this celebration. Thank you all for your kind support. I also request all your support for the grand success of our Institute Day celebration to be held today evening at our hospital wing. Thank you all.
Thank you, sir. Let me conclude with the words of science, technology, and innovation. Innovations that drive lasting economic growth emerge from the most advanced science, mathematics, and technology. May this year's Science Day celebrations inspire us to use science, technology, and innovation to get relief from the biggest pandemic ever. Let us all rise for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
So moving on to the next section of National Science Day celebration. Okay. So thank you so much, sir, for giving a details on the pandemic, and it was enlightening words for us. Most of the details which you shared with us, even I was not known to it. So thank you so much for giving us, beautiful presentation and enlightening us with same. Thank you. Now I request everyone to break for a tea. After that, we'll come back and then we'll move on to the next section. Thank you. Hello. So tea is arranged just behind the stage. So where the people are, you can see they're gathering around. I request everyone to go over there and have a tea and come back. Yeah. So I request all the students and colleagues to come forward. It's going to be interactive session. So probably you can move your chairs a little forward. Otherwise, we have chairs empty in the front. People can move front. Yeah, I request people to move front, please. Come forward. There are vacant seats. You can occupy these chairs. Request. Yeah. Yeah, right. Basically based on the slides. Hello. So everyone, welcome back. Now moving on to the next section of National Science Day celebration. I welcome Dr. Harikrishna S., Professor Cardiology Department, to enlighten us with his words on Science Day. Thank you, sir. Over to you.
Good morning. When you come forward, little bit because it is based on a PowerPoint. So if you can come forward, you can see it better. I will not ask any questions. Don't worry. Please come forward. Veilana. Okay. Okay. So Science Day has already been introduced. I'm not going to those details. Told multiple times. So I will ask the CCMB director has said. I will also discuss this today's Science Day based on the 1 year of COVID-19, how we tackled as a scientific community, how we tackled the COVID-19. So in December 2019, the first case of it was reported there is a cluster of cases of pneumonia in Wuhan. And the 1st January 2019, WHO set up a committee to assess the pandemic. And at that time, we were totally clueless. We didn't have any idea what is the nature of the virus, why certain people are badly affected, how to treat it, which drugs are effective, and how to prevent. We didn't have any clue on any of these things. We were worried. As a healthcare worker, I am seeing patients daily. I was worried when the mortality from Italy was 20%. 1 out of 5 patients were dying. So we were worried. And we saw the mass funerals in Italy. This is what at that time Francis Collins, president of NIH, said. This is a pandemic that is taking lives and destroying economies. And we have to do science in ways that people think we could not, driven by this sense of urgency. So there was an urgency in the scientific community. That is how the thing started. So first we identified the virus. It was an RNA virus, characterized. And this is what we have discussed, the spike protein, that red thing is a spike protein, very important component of the virus. That spike protein goes and attaches to the cell, the ACE receptor, that is some receptor in the human cell. Then came the big hype of how to prevent it. Hydroxychloroquine, that was the buzzword at that time. We had the maximum manufacturing capability of hydroxychloroquine, there was request from US, and there are some studies which was come up at that time showing the efficacy of hydroxychloroquine, and the world was looking towards India to get some export of hydroxychloroquine. But subsequently, I will show you how science works. It comes to a scientific study, we go for the ethics committee clearance, we do the study, comes out with the results, we do present it in meetings, submit for a publication, and we do something called a peer review. That is the more essence of science, peer review, because the people who have the same expertise, who have the same level of knowledge will assess that results and find out whether that is the results are acceptable. That is how it comes to the public domain. So chloroquine, in the situation of chloroquine also same thing happened. Chloroquine was not found to be effective, and many of the papers were retracted, and chloroquine was no longer a favored drug for prevention. Then we came to know that whenever a virus enters our body or any microorganism enters our body, the body produces antibodies against the microorganism. So actually the antibodies were hyper functioning. They were acting against our tissues. That is what is called a cytokine storm. We identified a cytokine storm, and we thought of ways to prevent or control the cytokine storm. We thought of drugs, and we started using steroids. Steroids are the drugs which is commonly available, very cheap drugs. We can use steroids to suppress the cytokine. So that is how we tried to control the mortality. We were partly successful. Then we thought of patients who have recovered from COVID-19. What do they have in their body? They have antibodies. So we thought of why can't we get those antibodies and inject it into patients so that they can control their infection? That is what the convalescent plasma. But somehow, due to many factors, which are human biology is a very complex system, you don't know. So because of the complexities, the data was not very successful. Then antiviral drugs. So many oseltamivir, which we are using for our routine viral fever, lopinavir, ritonavir, favipiravir, remdesivir, all these were tried. And this was the first combination tried, because when we sent our patients to medical college, all these patients received this combination. But subsequently, we got the evidence that it was not useful. And favipiravir came when we also, many of us tried to get some favipiravir and get in stock, because we are going to be exposed to patients. But subsequently, it also found to be not to be effective. Remdesivir, a big hype, 1,000 patient study was published. US FDA approved the drug. But in November 9, 2020, when we got more information that the drug is not effective, WHO said not to use it. And recently, the final word has come, it has only questionable benefit. That is how science is evolving. We cannot say a fact at a particular point, we may get better information in future. Then the big issue of vaccines, as the CCMB director has said. There are three main approaches of vaccine making. You can take the whole vaccine, kill it, the virus, kill it and use it. It will produce antibodies when you inject it. Or you can select the spike protein, take the spike protein and inject it. It will develop antibodies. Or you can take the genetic material like mRNA or whatever genetic material and inject it. That will produce antibodies. These are the different methods to produce them. And you should, this is a very important slide. You should see that how many years it will take to develop a vaccine normally. You can see that in the preclinical phase, 3 to 8 years it will take to develop the prototype of the vaccine. It will take another 2 to 10 years to develop the phase 1, 2, 3 studies. You should know what is phase 1, 2, 3 studies, because there are a lot of controversy in the news. Phase 1 is study, you try it in 10 patients to see whether it is safe. Second is phase 2, safety and immunogenicity in 100 patients. Phase 3, 1,000 patients. That is what they said about Covaxin. That 1,000 patients trial is going on. And it will take around minimum 5 to 18 years for a vaccine to come out to a phase 3 trial. That is what we are trying to do it in 5 months or 6 months or 1 year. So that is where science has grown, science has matured in this era in this 21st century. So many vaccines have been developed, 192 vaccines have been tried, and many of them are, 11 vaccines have been approved, including the vaccine which we developed with ICMR, the Covaxin. And India, we know that India is the vaccine hub of the world, because many of the 90% of the measles vaccine in the world is manufactured in India. So India is the vaccine hub of the world. and we have donated 22 million vaccines, 22 crore vaccines free, many of them free to 20 countries in the world. So this is what is called a vaccine diplomacy. Is the vaccine effective? Is the vaccine effective? This is the data. This shows that this is from Israel. Israel is a country where the technology is so advanced, they started vaccinating their population. And you can see that when the second dose vaccine has begun, you can see a sudden drop in the graph showing effectiveness of the vaccine. Today, I was very happy to, before that, Israel, what is the population of Kerala? What is the population of Kerala? Anybody? 3.3 crores or 3.4 crores as of now. So 3.4 crores, and India population is around 140 plus crores. So Israel has a population of 90 lakhs. They have already vaccinated around 37% of the population as of, even though India is vaccinated only 0.02% of the population, it is a huge task which we have done with our infrastructure. And Israel has already reopened the economy because of the vaccine success. So vaccine is effective. So our science has already proved victorious. And today morning, I was very happy to read this data which came in Hindu this morning. You can see that cases among health staff coming down from Kerala, our own Kerala. You can see that once the vaccination has started, even with a single dose, there is a significant drop in the number of healthcare workers becoming positive. So vaccine has been found to be effective. It is shown to be 98% protection against death due to COVID and 70% infection, protection against infection, protect some form of infection. And we are worried about the transition, global transition, as our CCMB director has already said. The South African variant, the UK variant of B117, all this, but we are not worried. The South African variant specific vaccine is ready for testing. Moderna has said the day before yesterday that our science has progressed so much that we should have faith in our science and scientists so that we can tackle all these problems. We have to maintain the cold chain. We should have the cold chain. What is cold chain? Whenever a vaccine is manufactured, just before it is injected to you, it has to be maintained at a particular temperature. The temperature for two of our vaccines, the Covishield and Covaxin, is 2 to 8 degrees Celsius. The other vaccines, some of the vaccines needs minus 80 degrees Celsius. That is why that infrastructure is not there in a huge country like India. That is why we selected these two vaccines, which can be stored in even a common refrigerator in your house. And today is a historic day where we are starting the vaccination for our elderly and the high-risk people above 45. So COVID-19 was a learning process for us. So we learned that high-risk patients, we isolated them, we gave care to them. Hyperimmune response, we found steroids are effective. Chloroquine was not useful in prevention. Antiviral drug remdesivir was not effective. Trials were done faster, regulatory approvals were faster, scientific community was given open access to their publications, and vaccines. There are a lot of questions to be unanswered. Which vaccine is the best? There are a lot of discussions on that. How long will the immunity last? When you will require the next booster? 6 months, 1 year, we don't know. I think we are sure that we will get the answers from the scientific community. So science continues to evolve. We realize our mistakes and correct it, as we have seen in the case of COVID. Chloroquine antivirus, we have seen, we initially said it was good, but subsequent data showed that it was not that effective. We changed our strategy. That is how science is evolving. It is a part of evolution and growth of science and aim to better. So that is it. So I will go on to tell you two stories about vaccination before I go to the next second part of my talk. So the story is about, I know that many of you must have known this story. The story of a milkmaid, a boy, a cow called Blossom, and a scientist. Somebody can tell whom I am speaking about? Okay. This is about who invented vaccine? Edward Jenner. This was about Edward. So Edward Jenner in 1796 in England, he was looking at that is what I said. Science is observing. Observation is science. So he observed that milkmaids never get smallpox, but he found that all of them have small scars of cowpox. Cowpox is a disease similar to smallpox. So all of them have these scars of cowpox in their hand, and they never get smallpox. So he observed and he did an experiment. So he inoculated pus taken from the pustule of the hand of a milkmaid and injected into a boy called James Phipps, his gardener's son. Whether it is ethical, it is a debatable question, but that proved, I think we are many of the people are leaving because of the success of vaccines. So James developed cowpox pustules. Because of the infection, he developed pustules, but he didn't have any. Cowpox is a very small, minor infection, just some pustules, and there is no systemic infection. So, but then what he did was Jenner got the smallpox pustules and injected it into James Phipps. That is called variolation, not injection actually, it is rubbing. And fortunately, this boy did not develop disease. So this was the first vaccination. So a disease with a 20% mortality. This was, you should know that this was in 1796. That was in 1800. So how many, 220 years back, when the science is in the very, very initial stage. Also, you should know about this long journey, 1802 to 1806, long journey. I think many of you must have read. These are all, what is the connection of this South American region with European countries? They all have their colonies, especially Spain. Spanish colonies are there in South America. So all these colonies, the people in the colonies developed smallpox, and so many people were dying. And in the royal families of Spain also many people died due to smallpox. And then this vaccination came, and King Charles VI of Spain sent an expedition to this South America by in the leadership of Dr. Bamis. But how will you take this virus from Europe to South America? That is a problem. There is no cold chain, nothing like that at that time. So they developed a mechanism. That is what science, I said, the way we have the human, we have evolved over these years. So what they identified 18 orphans from one of the hospitals and inoculated the first orphan with a cowpox pustule. And once that is going to heal, it will be transferred to the second orphan. So by transferring this infection to 18 people, they reached South America and started successful vaccination at that point. And this is the memorial of that orphan children, which is there in that South American region. And here also in Trivandrum also we have certain examples of the administrators taking very good examples, making good examples to spread science. In 1813, the smallpox vaccination came to Kerala, in Trivandrum, in Travancore. So people were very reluctant. Then His Highness Gauri Lakshmi Bai went with her family to General Hospital and took the vaccination. So that is how administrators can develop in the progression of help in the progression of science. I will show a few slides about future medicine and I will stop. So this is one area which is evolving in medicine. So you and your friend, you go everywhere together, have the same food, but your friend is very lean and healthy, looks healthy, and you are becoming obese every day. So you must have wondered what is happening. You will say, oh, his family is all obese, you can very lean, you can say and escape. But that is not. What is the reason? The reason is the gut microbiome. We have experiments to show that the bacteria which is reside in your colon or stomach is the reason for this difference. So they did twin experiments. Identical twins. One is obese, one is lean. They took the microbial flora from the gut of these two people and injected it into mice and gave it to the mice. And they found that those who had a lean body mass, the mouse did not become obese. But the mouse which received the microbiota from the obese person developed obesity. So this is what is seen. And it is linked to heart diseases. It is linked to neurological diseases, including autism. It has been found. I think in the future, we may be finding ways to alter the gut microbiome. And even cancer has been linked. For example, pancreatic cancer, the long-term survivors have a particular type of microbiome, while the patient who died faster have another form of microbiome. So can we alter the gut microbiome? We can do so many things. By taking a Mediterranean diet or a healthy diet, you can have a better microbiome. If you take so much red meat, your microbiome will be changed and you can develop an obese microbiome phenotype. Probiotics are available. You can take different types of probiotics to alter the microbiome, but these are all in the preliminary stages. You can the fecal transplantation is going to come in a big way for many diseases. Already it is in use for some infections. Now, so what do you use is get the donor feces, which is having good microbiota, filter it. Either you can introduce through endoscopy or you can make it into capsules and take it to alter the gut microbiome in a positive way. So Hippocrates in 460 BC has said all diseases begin in the gut. Probably we are looking into that now. Next is personalized medicine. Personalized medicine. So what is the current thing? For a particular disease, we give one drug for everyone. One drug for everyone. But what is happening? There will be some optimal responders. Some people will partially respond. Some will not respond. And if we can do something and find out which are the person who is responding, then we can give the drugs to that particular person. Or we can choose another drug which is found to be effective. This is what is called personalized medicine, basically based on genetic composition. So we have done a study in our hospital where we found that people who have artificial valves will require a medicine to prevent blood clotting. So we found a mutation where one group of people needed a dose of 7 milligram, but another group of patients required a larger milligram, up to 11 milligram. So if we can identify a genetic mutation, we can start the patient from what we do is we do a trial and error. We start with 2 milligram, go to 7, 11 like that. If we know the genotype, we can start from the beginning, the larger dose and make these patients reduce the hospital stay. So this is about precision medicine and personalized medicine, and everything will require big data analysis. Lot of huge number of personalized data you collect, process it, and develop it into pathways so that you can modify the treatment protocols, the diagnostic protocols, so that you are more targeted rather than a holistic approach. Cutting edge CRISPR gene editing, you must have read or you must have learned in your CRISPR gene editing. That has been useful in cancer therapy. Because it has been shown that they will use CRISPR gene editing to induce viral genome containing the adequate genes you want, and that can induce death of the cancer cells. 3D printed organs, that is going to be a future. I think we have a 3D printing facility here. So in the future, 3D printed organs will come. Initially, they will create a scaffold and they will create seed the cells over that, and you can heart and all is a big ask now because it is a so complex organ. So, but that will be the future. When you have a dilated cardiomyopathy or where the heart is damaged, you can get a 3D printed heart. That will be probably in another next 100 years. Distance care, telemedicine is another thing which is going to come. We know that telemedicine has been used in this COVID pandemic. Many of our patients in the peak of the pandemic, they were consulted through teleconsultations, video consultations, and even remote robotic surgery is possible. The surgeon with a high expertise is sitting in US and they can do operation on our patients in our operation table. That is possible. And virtual reality, that is the one which is going to come. Virtual reality, for example, to study anatomy, you need not go and dissect human bodies. You can take layer by layer dissections, and you can study the, I think virtual frogs and all, I think you must have, I don't know whether people are using it, but that is available. So science, I'm stopping here. Science is going to be fascinating. It's always evolving. Today's gospel truth may be totally discarded tomorrow. So that is what I am going to stress. It is all observation, changing the perceptions when you get more and more data. See, keep an open mind, question yourself. Why can't this be like that? Thank you.
Thank you, sir, for detailed overview on COVID virus and the use of science and technology to prevent it. Yes. We also thank you for sharing science stories and sharing future technology with us. Now it's open for question. Are there any question on this particular topic from audience?
Any questions? Yes, you're going to ask something. So everyone has understood everything. So I am very happy.
will make a comment upon. You see, it's very interesting to see that, as he said about COVID, it came into bats, but the bats don't have the disease. And they've been surviving with this coronavirus for such a long time, and it is co-speciating. It's sort of a big question as to how that happens. If we find out that very specific answer, we might actually find the solution. Because I'll tell you some of the reasons. It's probably having repressive genes which prevent inflammation. So you can get infected, but you don't start developing inflammation. And that's what he said. As to inflammation is a basic problem of all this COVID. But you see, the bat virus over a course of years have not produced an infection.
Okay, thank you.
Thank you so much, sir. Now it's time for some science quiz on Science Day. I welcome Dr. Ranjit S., Scientist B, Central Analytical Facility, as a quiz master. Ranjit, over to you.
Hello? Hello? നമസ്കാരം. സയൻസ് ക്വിസിലേക്ക് എല്ലാവർക്കും സ്വാഗതം. ഇതുവരെ നിങ്ങൾ ഒന്നും മിണ്ടാതെ ഇരിക്കുകയായിരുന്നു. ഇനി ആക്ച്വലി നിങ്ങളാണ് പാർട്ടിസിപ്പേറ്റ് ചെയ്യേണ്ടത്. അതുകൊണ്ട് നിങ്ങളുടെ റെസ്പോൺസ് വേണ്ട ഒരു ഘട്ടത്തിലേക്കാണ് നമ്മൾ എത്തിയിരിക്കുന്നത്. ആക്ച്വലി ഈ പറയുന്ന പോലെ ഒരുപാട് ക്വസ്റ്റ്യനൊക്കെ ചോദിച്ചിട്ട് നിങ്ങളിൽ നിന്ന് ആൻസർ വാങ്ങിക്കാനുള്ള സമയം ഇല്ലാത്തത് കൊണ്ട് തന്നെ ജസ്റ്റ് ഒരു റൗണ്ടിലേക്ക് നമ്മുടെ ഈ ക്വിസ് കോമ്പറ്റീഷൻ ചുരുക്കിയിരിക്കുകയാണ്. വളരെ സിമ്പിൾ ആയിട്ടുള്ള ക്വസ്റ്റ്യൻസ് ആണ്, ആർക്ക് വേണമെങ്കിലും ആൻസർ ചെയ്യാൻ പറ്റുന്നതാണ്. നിങ്ങളുടെ കയ്യിൽ എല്ലാവരുടെ കയ്യിൽ പേപ്പറും പേനയൊക്കെ ഉണ്ടല്ലോ, ഒന്ന് എടുത്തോളൂ, വേഗമാവട്ടെ. അതെ. ഓക്കെ, ആക്ച്വലി നമ്മുടെ റൗണ്ടിന്റെ പേര് ജിക്സോ റൗണ്ട് എന്നാണ്. ഓക്കെ, ആക്ച്വലി ഇവിടെ എന്താണ് നടക്കാൻ പോകുന്നതെന്ന് പറഞ്ഞാൽ ചില ശാസ്ത്രജ്ഞന്മാരുടെ പേര് ഞാൻ സ്ക്രാമ്പിൾ ചെയ്തിട്ടിട്ടുണ്ട്. അതായത് ലെറ്റർ അങ്ങോട്ടും റെഡിയാണോ? ഇങ്ങനെ മറുപടി വേണം. ഓക്കെ, തുടങ്ങാം. ഓക്കെ, ഫസ്റ്റ് ക്വസ്റ്റ്യൻ കാണുന്നുണ്ടോ? പെഗ് സാങ് വിത്ത് ഹെൻ. ഒന്ന് നോക്കാമോ? കറക്റ്റ് ആൻസർ സ്റ്റീഫൻ ഹോക്കിംഗ്. ഒരു സമ്മാനം കൊടുക്കൂ. പേര് പറയൂ. അനിത. അനിക. ഓക്കെ, കൺഗ്രാറ്റ്സ്. സോ ആക്ച്വലി കുറച്ച് ക്ലൂസ് ഒക്കെ ഇട്ടിട്ടുണ്ടായിരുന്നു. ഫസ്റ്റിലെ ആൻസർ പറഞ്ഞില്ല എങ്കിൽ അവർക്ക് വേണ്ടിയിട്ട് കുറച്ച് ക്ലൂസ് ഇട്ടിട്ടുണ്ടായിരുന്നു. ഫസ്റ്റ് ക്ലൂ ഇതാണ്, ബ്ലാക്ക് ഹോൾ. ഹിസ്റ്ററി ആൻഡ് ടൈം, ബ്രീഫ് ഹിസ്റ്ററി ഓഫ് ടൈം. So that is Stephen Hawking. Next question. One mark. Simple. Any idea? Clues ആണോ? CV Raman. Okay, correct answer. Actually 2-3 പേരെ എണീറ്റിട്ടുണ്ടായിരുന്നു. ഓക്കെ, ഒരാൾക്ക് സമ്മാനം കിട്ടിയതുകൊണ്ട് അടുത്ത ആൾക്ക് കൊടുത്താലും മതി. ഓക്കെ, നെക്സ്റ്റ്. സോ ഇതിന് ഉണ്ടായിരുന്ന ക്ലൂസ്, സോ യു ആൾ നോ, സിയുടെയും ആകാശത്തിന്റെയും നിറത്തിന് കാരണം രാമൻ എഫക്റ്റ് ആണെന്ന് പറഞ്ഞിട്ടുണ്ട്. ദെൻ ഭാരതരത്നയും നോബൽ പ്രൈസും നേടിയ ആളാണ് CV രാമൻ, ചന്ദ്രശേഖര വെങ്കിട്ടരാമൻ. ഓക്കെ, next question. Interest in ELBA. ആൽബർട്ട് ഐൻസ്റ്റീൻ. കറക്റ്റ് ആൻസർ. അതെ അതെ, ആൻസർ പറയുന്ന ഒരു കൈ ഉയർത്തി പേര് പറഞ്ഞിട്ട് സമ്മാനം മേടിക്കുക. ഇൻട്രസ്റ്റ് ഇൻ എൽബ. സോ ഇതൊക്കെയായിരുന്നു ക്ലൂസ്. 1921 ഫിസിക്സിന് നോബൽ പ്രൈസ് കിട്ടി. എന്തിനാണ് കിട്ടിയത്? തിയറി ഓഫ് റിലേറ്റിവിറ്റിക്കാണോ? അല്ല, ഫോട്ടോ ഇലക്ട്രിക് എഫക്റ്റിന്റെ എക്സ്പ്ലനേഷൻ കൊടുത്തതിനാണ് 1921 നോബൽ പ്രൈസ് കിട്ടിയത്. പിന്നെന്താ, ഇത് എല്ലാവർക്കും അറിയുന്ന ക്ലൂ ആണ് അല്ലേ? ഓക്കെ, സോ ഇറ്റ് ഈസ് ആൽബർട്ട് ഐൻസ്റ്റീൻ. നെക്സ്റ്റ് ക്വസ്റ്റ്യൻ. ജനുവരി ഈസ് സ്വർണ്ണമാൻ. ജനുവരി സ്വർണ്ണമാനാണോ? പ്ലീസ്. ശ്രീനിവാസ രാമാനുജൻ. കറക്റ്റ് ആൻസർ. സോ ഇതൊക്കെയായിരുന്നു ക്ലൂസ്. സോ ഈ ഫിലിം കണ്ടിട്ടുണ്ടോ? ദ മാൻ ഹൂ ന്യൂ ഇൻഫിനിറ്റി. രാമാനുജന്റെ ലൈഫിനെ ആസ്പദമാക്കിയിട്ട് വന്ന ഫിലിമാണ്. ദെൻ ഈ ടാക്സി ക്യാബിന്റെ നമ്പർ ഓർമ്മയുണ്ടോ? 1729. ഓക്കെ, ദാറ്റ് ഈസ് രാമാനുജൻ നമ്പർ. നെക്സ്റ്റ് ക്വസ്റ്റ്യൻ. ഹരിയാന ബോൺ ഗ്രോക്ക്. പ്ലീസ്. ഹർഗോബിന്ദ് ഖൊറാന. കറക്റ്റ് ആൻസർ. നിങ്ങളെല്ലാം ബ്രില്യൻ്റ് ആയതുകൊണ്ട് പെട്ടെന്ന് പരിപാടി ഇപ്പൊ കഴിയും കേട്ടോ. ഓക്കെ, ഇത് ഹർഗോബിന്ദ് ഖൊറാനയുടെ 96th ബർത്ത്ഡേക്ക് വേണ്ടിയിട്ട് ഗൂഗിൾ കൊണ്ടുവന്ന ഡൂഡിലാണ്. ഇത് പുള്ളിക്ക് 1968 നോബൽ പ്രൈസ് കിട്ടിയിട്ടുണ്ടായിരുന്നു. അതായത് DNA യുടെ ജനറ്റിക് ലാംഗ്വേജ് കോഡിംഗ് അറേഞ്ച്മെന്റ് കണ്ടുപിടിച്ചതിന്, അല്ലെങ്കിൽ അതിന് വേണ്ടിയിട്ട് നിറൻബർഗുമായിട്ട് ചേർന്ന് പുള്ളിക്ക് നോബൽ പ്രൈസ് കിട്ടിയിട്ടുണ്ടായിരുന്നു. ഓക്കെ, സോ ദാറ്റ് ഈസ് ഹർഗോബിന്ദ് ഖൊറാന. നെക്സ്റ്റ് ക്വസ്റ്റ്യൻ. മെത്തേഡ്സ് ഓൺ സലൈവ. തോമസ് ആൽവ എഡിസൺ. കറക്റ്റ് ആൻസർ. ഇതൊക്കെയാണ് ക്ലൂസ്. വിസാർഡ് ഓഫ് മെൻലോ പാർക്ക്. മെൻലോ പാർക്കിലെ മാന്ത്രികൻ എന്നറിയപ്പെട്ടിരുന്നത് ആരായിരുന്നു? തോമസ് ആൽവ എഡിസൺ ആയിരുന്നു. പുള്ളിയുടെ ബൾബ് കണ്ടുപിടിച്ചത് പുള്ളിയാണ്, തോമസ് ആൽവ എഡിസൺ. ഓക്കെ, നെക്സ്റ്റ് ക്വസ്റ്റ്യൻ. ഐ ആം റൂറി. മേരി ക്യൂറി. കറക്റ്റ് ആൻസർ. ഓക്കെ, ആദ്യം പറഞ്ഞതുപോലെ ഈ പ്രോഗ്രാമിന്റെ തുടക്കത്തിൽ പറഞ്ഞതുപോലെ 2 വ്യത്യസ്ത ശാസ്ത്രശാഖകൾക്ക്, അതായത് ഫിസിക്സിനും കെമിസ്ട്രിക്കും നോബൽ പ്രൈസ് നേടിയ ആദ്യത്തെ വനിതകളിൽ ആദ്യത്തെ ആളാണ് ആര്? മേരി ക്യൂറി എന്ന് പറയുന്നത്. വേറെ ആർക്കെങ്കിലും കിട്ടിയിട്ടുണ്ടോ നോബൽ പ്രൈസ് 2 തവണ? കെമിസ്ട്രിക്ക് ആർക്കാ കിട്ടിയ 2 നോബൽ പ്രൈസ്? ഫ്രെഡറിക് സാങ്ങർ എന്ന് കേട്ടിട്ടുണ്ടോ? ഫിസിക്സിന് ആർക്കെങ്കിലും കിട്ടിയിട്ടുണ്ടോ? ജോൺ ബാഡീൻ എന്ന് കേട്ടിട്ടുണ്ടോ? ഓക്കെ, ഇനി ലീനസ് പോളിങ്ങിന് കിട്ടിയിട്ടുണ്ടോ? കിട്ടിയിട്ടുണ്ട്. പുള്ളിക്ക് പീസിനും അതുപോലെ തന്നെ കെമിസ്ട്രിക്കും കിട്ടിയിട്ട് നോബൽ പ്രൈസ് കിട്ടിയ വ്യക്തിയാണ് ലീനസ് പോളിംഗ്. ഓക്കെ, നെക്സ്റ്റ് ക്ലൂ ഇതായിരുന്നു. റേഡിയം, പൊളോണിയം എന്നീ എലമെന്റ്സ് ആരുടെ സംഭാവനയാണ്? മേരി ക്യൂറിയുടെ സംഭാവനയാണ്. ഓക്കെ, നെക്സ്റ്റ് ക്വസ്റ്റ്യൻ. ഹാൾട്ടൺ ജോൺ. ജോൺ ഡാൾട്ടൺ. കറക്റ്റ് ആൻസർ. അപ്പൊ ഒരുപാട് പേര് പറഞ്ഞു. ഓക്കെ, ആറ്റം തിയറി 1808 ഒരു ബ്രിട്ടീഷ് സ്കൂൾ ടീച്ചർ ആയിരുന്ന ജോൺ ഡാൾട്ടൺ ആണ് പ്രപ്പോസ് ചെയ്തത് അല്ലേ? അതുപോലെ തന്നെ ഡാൾട്ടനിസം എന്ന് കേട്ടിട്ടുണ്ടോ? കളർ ബ്ലൈൻഡ്നെസ്, അവിടെയും ഡാൾട്ടന്റെ പേരുമായിട്ട് ബന്ധപ്പെട്ടിട്ടാണ് വരുന്നത്. സോ ജോൺ ഡാൾട്ടൺ. നെക്സ്റ്റ്, സ്കാർ ഹീൽ വിന്റർ. പറയൂ. Charles Darwin, correct answer. So ഇതൊക്കെയായിരുന്നു ക്ലോസ്. ഡാർവിൻ സ്വിഞ്ചസ്, HMS ബീഗിൾ, ചാൾസ് ഡാർവിൻ. ഓക്കെ, എത്രയായി ക്വസ്റ്റ്യൻസ്? 10 ആയോ? ഇല്ല. സല്യൂട്ട് അവർ സിപ്പ്. എനി ഐഡിയ? ക്ലൂ വേണോ? വേണോ? ഫസ്റ്റ് ക്ലൂ, ലൂയി പാസ്റ്റർ, കറക്റ്റ് ആൻസർ. It is Louis Pasteur. ഓക്കെ, താങ്ക് യു. പരിപാടി കഴിഞ്ഞു. എല്ലാവർക്കും നന്ദി, നമസ്കാരം.
Thank you, Dr. Ranjit. It was really entertaining. Now let's have some fun with science magic. I welcome Dr. Jijo Raj, Scientist C, for a visual treat on science magic.
Thank you, the organizers, and thank you all for coming. Today we shall see some science experiments which I hope you will enjoy. With me is Jijo, the second, and we will do some tricks. The idea is to use simple things that you can find in your home, and with these things you can try to demonstrate some nice things that explain some principles of science. First is a water fountain that works perpetually, automatically, without any source of energy. So first we'll put some water. We need a reservoir of water. It's better to put some color so that you can enjoy some. Yes. This is a bottle, a Fanta bottle that we usually have, and I fill it with a little amount of water, just a little bit, maybe a little bit more, and then I close it. Okay, I have to close very tightly, and the trick is I'll just put it in this reservoir. And then you can see a fountain working without a source of electricity, nothing. Could you see? and shooting it up. And if you have an opportunity to visit Geneva, in the right in that lake Geneva, you have a similar structure, which is called water jet below. That means water jet, which was it's almost like it's a miniature of. Thank you. And we'll see the next. I'm not so sure if everybody can see this, but this is a paint can, and this paint can likes to move not downwards but upwards. He don't want to go down. He always want to go up. And why is that so? Excuse me. Please say. Okay. So it's a principle about center of gravity. So I have glued a mass that is off center, and even though it is, we put it, the tin can rolls to its center of gravity position. So that is the trick there. Thank you. And how many of you have this fish in your like a pet fish in your home? After the lockdown, I have seen that many people turn to have aquariums in your home. How many of nobody like aquarium? Okay. So in aquarium, what is important for fish is to have air, and the air you need to have a small motor that make this air bubbles. Now here we demonstrate a simple technique where you can get some air bubbles. Once again, this is a Fanta bottle. I just fill it with water. See that? You see? It's continuously making bubbles by itself and without any electricity. This is aeration. Oxygenation, important for many chemical processes, important for life in aquatic systems, all that. So the technique is quite simple. We actually, you have an outlet at the bottom, and just above the outlet you have another refill which is inserted onto it, and that is at a slanted angle, and when water flows through that, there is a pressure drop, and this pressure drop is actually sort of, have you heard about cavitation effect? when when the fluid medium, pressure of the fluid medium decreases, its boiling point, it boils actually. So there will be boiling, and it's not exactly cavitation, but similar principles. And this pressure difference, the decrease in the pressure actually drives this air from the up and makes these bubbles. Well, this can be any model of a planet or an atom. And in atom, what happens? What is the structure of atom? You have a nucleus, and then you have orbits around, and the electrons revolve around this orbit. That's the same at the fundamental part of matter, the atom. And also it's the same sort of model in the global universe, where you have the solar system with the sun, the earth are revolving around this orbit. I should catch it closely. Yeah. Goes by its own. So yeah, that freaking noise is because of the magnet here. There is a magnet which is rotating around this rails. So this is the planetary motion. Okay. I think we can still see show some walking capsule. This is a capsule. I mean, sort of a capsule medicine. But this guy likes to go for a walk by its own. Okay. Okay. So the idea here is we have a mass, a spherical mass inside, and that is creating this this sort of like walking effect. We know the color white light. White light is composed of primary colors, blue, yellow, and red, like that. Okay. And we try to just mix them. It's white, right? Okay. Anybody can volunteer? Yes, please. On the center. Okay. That's enough. I mean, so this shows that how hard you hit it, it doesn't break. But you hit it, it breaks. So this is the strength of the unity. Okay, if we stand together, we can face great challenges in life, our country, everything. And even this corona crisis, we can face it successfully if we stay together. Thanks a lot for your volunteering. Thank you. And that's this message. Thanks to our unity, this corona virus, we can put the trap here. Okay, we can trap it. Thank you very much. Have a nice evening. Have a great day, Science Day. Please enjoy our campus, our science we do here. All the best for you. Thank you very much. Thank you, Jijo.
Thank you, Jijo. It was really amazing. So thank you all once again, and special thanks to students and faculty from Government Women's College Trivandrum for their presence. Now we close the program and get ready for lab visits. Students are divided in teams and volunteers will lead them to their respective labs. You're also welcome for lunch arranged in the canteen annex. We request students to maintain social distancing norms during lab visits.
So there will be 8 teams.